Hi, and welcome back to Chapter 14. We are continuing our discussion of antibacterial drugs, and now we're going to go on to inhibitors of metabolic pathways. So let's look at the last antibacterial target, which are metabolic pathways. So some drugs mimic normal cellular substrates and compete for enzyme binding sites. For instance, if I look down here, I have an enzyme, which is this gray structure, my normal substrate in the metabolic pathway, and then here is my drug or inhibitor. So you can see the shape down here is very similar between the substrate and inhibitor, and what the drug can do is bind to the enzyme at the site that the substrate normally does, and therefore blocks that pathway. So they block key steps like folic acid synthesis, which bacteria need to make nucleotides and DNA. And when those reactions stall, the cell cannot grow or divide. First, we have our folic acid inhibitors, and there are two major classes that work together here. We have our sulfonamides, such as sulfamethoxazole, and these mimic PABA, which is a precursor in the pathway that bacteria use to eventually make DNA and RNA. So these drugs look just like PABA, and they bind to and block the enzyme that makes folic acid. So when they bind here, nothing else will happen down the line. Trimethoprim is similar, and this blocks the next enzyme that converts dihydrofolic acid to tetrahydrofolic acid. So it blocks this step right here. And they are often used in combination. So trimethoprim is sometimes abbreviated TMP, and sulfamethoxazole is sometimes abbreviated SMX. When they are combined together, this is usually marketed as a drug called Bactrim, and they together completely shut down folate synthesis. So this is a synergistic bactericidal effect. And these drugs can be used to treat urinary, ear, and respiratory infections. And because human cells, we don't make folic acid. We get folic acid from our diet, so these drugs target bacteria selectively. Here's another look at that same pathway. So this is the normal pathway that's happening in bacteria. They have a compound called PABA, para-amino-benzoic acid. Don't worry about that. It's going to bind to an enzyme and create this intermediate dihydrofolic acid that binds to another enzyme to eventually form nucleotides. And sulfonamides look similar to PABA, so they will bind to the enzyme and block future steps. Trimethoprim looks kind of like dihydrofolic acid, so it'll bind to this enzyme and then block subsequent steps that way. How do sulfonamides and trimethoprim selectively target bacteria? So we just saw that both drugs are anti-metabolites that block steps in bacterial folic acid synthesis, and that was essential for the bacteria to make DNA and RNA. Sulfonamides mimic PABA, and don't worry about what it stands for, but if you're interested, PABA stands for para-amino-benzoic acid. And sulfonamides, therefore, block the first enzyme in that folate pathway. Folate is the same thing as folic acid. Trimethoprim blocks a later enzyme that converts dihydrofolate to tetrahydrofolate. And humans, we don't synthesize folic acid. We get it from our diet, so these drugs selectively inhibit bacterial metabolism without affecting human cells. And these two drugs, when they're combined, they have a synergistic bactericidal effect. Our next set of drugs inhibit mycolic acid and ATP synthase. We have two specialized agents that both target mycobacteria, including the bacterium that causes tuberculosis. Isoniazid inhibits or blocks mycolic acid synthesis that weakens that wax-like cell wall, and it's often combined with rifampin or streptomycin to prevent resistance, but it can also cause liver and nerve toxicity. Bedaquiline targets ATP synthase. This is the enzyme that makes energy for the cell, so this essentially cuts off the power supply to that cell. It's often used as a last resort tuberculosis drug because it can carry serious side effects such as liver damage or heart arrhythmias. And our book has a table here that focuses on major antimetabolite drugs like those that target folic acid synthesis or those that target mycolic acid synthesis. To recap some of what we've talked about in 14.3, there are several antibacterial targets of these drugs. They could attack the cell wall, protein synthesis, membranes, nucleic acids, or even metabolism, as we just saw. How well the drug targets the microbe without harming us, this is known as selective toxicity, depends on how different the microbial structure is from the human host cell equivalent structure. Combination therapy is often used where we combine drugs in order to make the treatment more effective and or lower the likelihood for resistance to the drug. But there is some toxicity risk with some of the drugs that have very serious side effects. So let's summarize the mechanisms of action of the different drug groups. First, we had the inhibition of cell wall biosynthesis, and these drugs included the beta-lactams, vancomycin, or bacitracin. These blocked enzymes that form peptidoglycan or the cross-links of peptidoglycan, or they prevented transport of the cell wall precursors outside of the membrane. So overall, the result was that it weakened the bacterial cell wall, leading to cell lysis and death. And these are selectively toxic because we do not have peptidoglycan in our human cells. For the second one, we have protein synthesis. These were drugs like aminoglycosides, tetracycline, macrolides, and several others. These bound to the 70S ribosome in bacteria. So we had that 30S subunit of the ribosome or 50S subunit, and they basically prevented the accurate reading of messenger RNA or transfer RNA binding or peptide bond formation. So overall, they block the production of critical bacterial proteins that can cause death of the cell. But in terms of selective toxicity, human ribosomes and we have 80S ribosomes while they have 70S. They're structurally different, but remember there can be side effects because we do have 70S ribosomes in our mitochondria. The third group were the membrane, drugs that affect membrane function, and these include the polymyxins. So these drugs insert themselves into bacterial membranes, they act like detergents, and they create holes in the membrane. That can lead to a loss of the cell contents and cell death, but they're limited in use because they can also damage our membranes. So these drugs are usually used topically or as a last resort drug. The next one are drugs that affect nucleic acid synthesis. So these were drugs like rifampin or ciprofloxacin. These block RNA polymerase so that there's no transcription, or they can block DNA gyrase or topoisomerase so the DNA cannot open or unwind for replication. So these overall prevent bacterial DNA or RNA synthesis, and in terms of selective toxicity, they target bacterial-specific enzymes that we don't have in our human cells. Lastly, the metabolic pathways we just talked about. So these include the sulfonamides, trimethoprim, isoniazid, and bedaquiline. So these mimic normal substrates. They can block folic acid or mycolic acid synthesis, and they basically prevent or block precursors for nucleic acid synthesis or cell components. And in terms of selective toxicity, humans, we get folic acid from our diet, but bacteria have to make it, so it's selectively toxic against those bacterial enzymes. Now we'll transition into 14.4, where we're going to look at drugs that affect other microbes, such as fungi, protozoa, helminths, and viruses. So beyond drugs that attack bacteria, those antibacterials, we also have drugs for fungi, protozoa, helminths, and viruses. But these infections are harder to treat because they are eukaryotic, just like us. For viral infections, remember they're not eukaryotes or prokaryotes, they're not alive. They pose their own challenge though because they replicate inside our cells and use our machinery. So the goal is to find targets that are unique to the pathogen while sparing and protecting human tissues. Let's start with our antifungal drugs. So these target structures or processes that are unique to fungi and not found in our cells. For instance, fungi contain ergosterol in their cell membranes instead of cholesterol, which is what we have. So cholesterol looks like this. That's what we have in our cell membranes to maintain membrane fluidity, whereas fungi have ergosterol, which is similar in structure but a different chemical compound. So most antifungals either block ergosterol synthesis or bind to ergosterol directly to disrupt the integrity of the cell membrane. Other drugs may affect the cell wall or interfere with mitosis, and the goal is to damage the fungal cell without harming human cells. We have two classes of ergosterol inhibitors, the azoles, like fluconazole or ketoconazole. These block enzymes that make ergosterol. We also have the allylamines, such as terbinafine, also brand name is Lamisil for that one. This blocks another enzyme in an earlier step in the ergosterol production pathway. The result of these drugs are weakened, leaky cell membranes, and these agents can treat skin, nail, and systemic fungal infections. Our next set of drugs bind to the membrane of fungi. So we have polyenes like nystatin or amphotericin B. They bind to ergosterol and form pores, these holes in the fungal membrane, leading to the leakage of ions and other cell contents. Eventually, the fungal cell dies. So these drugs are fungicidal, but can harm human cells at high doses. Amphotericin B is used for intravenously for serious systemic infections, and nystatin is used topically or orally for Candida yeast infections. Here's an example of the fungal cell membrane with ergosterol embedded for membrane fluidity. Amphotericin B binds to ergosterol in the membrane, forming these giant pores or holes in the membrane that causes ions and other cell contents to leak out that kills the fungal cell. We also have antifungals that attack the fungal cell wall or fungal cell division. This includes echinocandins such as caspofungin. This drug blocks beta-glucan synthesis. So this is a component of fungal cell walls, and we do not have cell walls, so we don't have beta-glucan. For nicomycin Z, this interferes with, again, the cell wall, chitin synthesis in fungi, but it's still experimental and not FDA approved. Griseofulvin acts on microtubules that are involved in mitosis or cell division in fungi, and we use this to treat dermatophyte infections like ringworm or athlete's foot. And dermatophytes in general, these are a group of fungi that infect our keratinized tissues like our skin, hair, and nails. So these cause superficial infections like I mentioned, ringworm, athlete's foot, or even jock itch. We also have antifungal antimetabolites. Remember, antimetabolites are drugs that mimic normal cell substrates, so they compete for enzyme binding sites. We have atovaquone. This blocks the electron transport chain in mitochondria and can affect both fungi and some protozoa. Flucytosine enters fungal cells and then it's converted inside the fungal cell to a toxic form that interferes with DNA and protein synthesis. These drugs are often used in combination to fight systemic infections, such as those caused by Cryptococcus neoformans in immunocompromised patients. So systemic infections are infections that have invaded our deep tissues and usually are spread throughout the body through the blood. Opportunistic infections are caused by bacteria that typically don't cause disease in healthy individuals, but can cause these opportunistic infections in immunocompromised patients. These drugs are also often given in combination to avoid resistance to the drug. This figure summarizes our targets of the antifungal drugs. Remember, in fungal cell membranes, they have ergosterol to maintain membrane fluidity, whereas we have cholesterol. So we had drugs like azoles or allylamines to inhibit ergosterol synthesis. We also had drugs that bound to the ergosterol to generate holes or pores in the membrane, and that would cause leakage of ions and other cell contents. We had groups of drugs that block the formation of critical cell wall components, such as beta-glucans or the chitin, and drugs that block normal cell function, such as inhibiting DNA or disrupting mitosis by blocking or disrupting microtubules. We even had drugs that affected the electron transport chain. And our book also has a table for our antifungal drugs. So to summarize, we had drugs that inhibited ergosterol synthesis, drugs that bind to ergosterol to form pores in the membrane, as well as drugs that inhibited cell wall synthesis by blocking beta-glucan or chitin synthesis, as well as drugs that block or inhibit mitosis by inhibiting microtubule formation. This checkpoint asks, how is disruption of ergosterol biosynthesis an effective mode of action for antifungals? So ergosterol is the main sterol in fungal cell membranes, and it serves the same structural role as cholesterol in human cells. Antifungal drugs like azoles or allylamines, they block enzymes involved in ergosterol synthesis. So without ergosterol, the fungal membrane becomes unstable, it becomes leaky, and it disrupts nutrient transport. So things might leak out of the cell, normal nutrients can't come in, this leads to cell death. And it's selectively toxic because human cells, we use cholesterol, not ergosterol, so we're usually not affected, although we can be affected if the drugs are being given at a very high dose. When the drugs are present in high concentration, they become less specific and can also affect cholesterol in human cells. Here our book provides us with a clinical case study on Jack, a 48-year-old engineer who's HIV positive but generally pretty healthy because of antiretroviral therapy or ART. But he has a tough time at work and develops a fever and dry cough. When he goes to the doctor, they determine that he has pneumocystis jirovecii pneumonia, and this formerly was known as pneumocystis carinii. So just in case you might have heard this name as well before. This is an opportunistic fungal pathogen. So normally pneumocystis pneumonia is not seen in healthy individuals, but it can be seen in immunocompromised patients such as those with HIV. So even though pneumocystis is fungal, it can actually respond to an antibacterial combination drug. We saw this earlier. Bactrim is also known as TMP-SMX. TMP stands for trimethoprim. SMX stands for sulfamethoxazole. And this is because this fungi, or this fungal organism, it depends on folate synthesis like bacteria do. So unlike humans, we get our folic acid from our diet. Fungi depends on or has to produce its own folic acid. So this really illustrates how drug mechanisms can kind of overlap between kingdoms. Even though this is generally an antibacterial drug, it's also effective against fungi. And down at the bottom, they have a smear of Jack's sputum sample, and they did direct immunofluorescence assay. So this is basically antibodies that bind to the pneumocystis fungi, and they're tagged with immunofluorescent antibodies. Next, we have our antiprotozoan drugs. Protozoa are single-celled eukaryotes with complex life cycles, and their drug targets include metabolism, DNA synthesis, and redox reactions. Remember redox? These refer to oxidation reduction processes. But because protozoan diseases vary so much, a lot of these drugs tend to be pathogen specific. We have drugs that specifically target malaria or giardia or amoebas, for instance. And some of these agents work against both protozoa and fungi due to overlap in their metabolism or structures. And here is a list of some examples of antiprotozoan drugs we have and their major mechanisms of action. So atavaquone blocks mitochondrial electron transport and thus stops ATP production. We also have artemisinin. This generates reactive oxygen species when it's activated by heme, and that will damage parasite proteins and the membrane of the protozoan. Pyrithamine and sulfadiazine, these both block folic acid synthesis. Metronidazole and tinidazole, these both damage DNA in anaerobic protozoa. Chloroquine, this one blocks heme detoxification in malaria parasites. So what happens is when the protozoa breaks down the hemoglobin in our red blood cells, it creates toxic heme intermediates, and then the protozoan has to break down the toxic intermediates. If this drug prevents that destruction of the toxic intermediates, then those intermediates will kill the parasite. And often we use these drugs in combination to slow resistance against the drug. Like the previous section, our book also has a table of antiprotozoan drugs, and take a look at their mechanism of actions. We can inhibit electron transport in mitochondria, inhibit that folic acid synthesis, make reactive oxygen species that can damage the cell, inhibit DNA synthesis, or inhibit heme detoxification. This checkpoint asks us to list 2 modes of action for antiprotozoan drugs. So antiprotozoan drugs can use several strategies depending on the organism, but 2 major modes are 1, to inhibit the electron transport chain or inhibit energy metabolism in general. We have that atavaquone. So atavaquone blocks the mitochondrial electron transport chain I didn't talk about this yet, but it blocks it in Plasmodium, which causes malaria. It can also block it in Toxoplasma. And another mode is interference with nucleic acid synthesis or DNA integrity. For example, metronidazole causes DNA strands to break in anaerobic protozoa, and this can be used to treat Giardia, Trichomonas, or Entamoeba. So some of these we'll see later in the semester and or in the lab as well. We also have other mechanisms that inhibit folic acid production or heme detoxification. So next we have our antihelminthic drugs. Remember that helminths are large multicellular parasitic worms with metabolism that's similar to ours, so finding safe targets is pretty hard. Most drugs will aim to either paralyze or starve the worm or block their energy production. So the drugs will exploit various neuromuscular and metabolic pathways that are not present in humans. Here are several examples of antihelminthic drugs. We have our benzimidols, such as mebendazole or albendazole. These block microtubule formation, especially in the intestinal cells, so now the worms cannot absorb glucose and they essentially starve to death. Here, ivermectin, this one disrupts nerve signaling, and that leads to paralysis and death. Niclosamide blocks ATP production in tapeworms specifically. Praziquantel, this one increases membrane calcium permeability, and that will lead to muscle spasms and paralysis in the worm. And all of these drugs selectively target functions that are absent in human cells, so they're selectively toxic. Here's that equivalent table for antihelminthic drugs. You can see the different categories or mechanisms of actions we discussed. Don't worry about drugs that I never mentioned, but this is a nice little reference to have in addition to the previous tables as we get into these topics in the laboratory. Why are antihelminthic drugs difficult to develop? So helminths are parasitic worms, and they're also multicellular eukaryotes like us. They are biologically similar to humans. They share many of the same metabolic pathways and the same structural pathways. So there aren't that many unique drug targets. They also have a large size and pretty complex life cycle, and that makes it even harder to find a safe, effective treatment. Of the drugs that do exist, they have to target worm-specific structures like specific microtubules or neuromuscular function that's only in the worms. But again, these can also still have toxic side effects in humans. So it's hard to get selective toxicity because here it's difficult to kill the parasite without harming the human host since we are so similar. Now we're going to switch gears and look at viruses and antiviral therapy. So we have several antiviral drugs, but viruses are a challenge. Since they use our own cellular machinery to replicate, there aren't that many unique targets. So the drugs that we do have, they have to act on virus-specific enzymes or virus-specific life cycle steps. But because viruses also mutate pretty quickly, resistance can be, unfortunately, pretty common. And that's why a lot of antivirals often work best in combination. For example, we have for HIV something called HAART therapy, which stands for Highly Active Antiretroviral Therapy. So we're going to see this later on in the section, but it's a combination of multiple antiviral drugs that target different steps in the HIV life cycle. One way we can attack viruses is by preventing their synthesis of nucleic acids, DNA or RNA. And we do this by using nucleoside analogs. These are drugs that look just like one of our normal nucleosides. So remember, a nucleoside is different from a nucleotide because it's missing the phosphate group. So normally we have a phosphate group attached to carbon number 5. Without that phosphate group, like the one shown here, it's called a nucleoside. And our normal nucleosides are adenosine, so that's A, guanosine, thymidine, and cytidine, so AGTC. But the analog is a chemical that looks kind of like one of our nucleosides, but when it's incorporated, it blocks the continuation of DNA replication or RNA formation. So it causes chain termination, kind of like when we were looking at sequencing DNA. There we had those dideoxynucleotides. That's a similar concept. Acyclovir, for instance, also known as Zovirax, acyclovir looks similar in structure to guanosine. So when the virus is trying to replicate its DNA, instead of putting down the guanine or guanosine, it might accidentally put down the drug, acyclovir. If it does that, that stops the synthesis of DNA, and so the virus is not able to successfully replicate itself. This one, for instance, is used to treat herpes viruses. We also have ribavirin, which is similar. It's also a nucleoside analog, but this one is used to treat or attack RNA viruses. Sofosbuvir, also known as Sovaldi, is a uridine analog. So remember, normally we have uracil in RNA, so uridine is the nucleoside, and this one blocks the RNA virus, hepatitis C. AZT, which stands for azithromycin zidobutene, this one inhibits reverse transcriptase in HIV and prevents the virus from turning its RNA into DNA. So that action is prevented. So all of these drugs will stop viral genome replication before they can make new virions. Here's an example of how those nucleoside analogs work. Acyclovir was an analog of guanosine. They look similar in structure. So if we are using acyclovir for treatment of, for instance, herpes, let's say acyclovir is being given, it gets into the cell, the viral enzymes will add a phosphate group, and then our enzymes will add additional phosphate groups to form acyclovir triphosphate inside the cell. Meanwhile, the virus is trying to replicate its genome. So here's its genome, and here's the new copy it's trying to make. It looks like the next nucleotide that's supposed to be added is complementary to C, so it should be G. It might incorporate the regular nucleoside or nucleotide in this case, but it might incorporate our drug, acyclovir. If it accidentally puts down acyclovir here, then the replication process will stop and the virus will not be able to replicate its genome. And that takes us to the end of part 3. In our next video, we'll finish our discussion of antiviral drugs.